Hello everyone, my name is Shadi Ainolahi Darabad and I'm going to present my study on the comparison of uricase enzyme production in clinical and environmental strains of Pseudomonas and Bacillus. Let's start by a brief introduction of uricase enzyme. Uricase is an oxidoreductase that catalyzes the oxidation of uric acid into allantoin, a more water-soluble compound which is easily excreted by the kidney. As you can see on this slide, uricase is produced by many living organisms such as bacteria, fungi, plants, and all mammals except humans and certain primates due to missense and frameshift mutations in the gene encoding the enzyme. Therefore, due to the absence of this enzyme in humans, the concentration of uric acid in plasma is rather high, and an abnormal rise of it can result in renal failure and gout. Uricase is also used in laboratory analysis for the detection of uric acid and in some cases as a therapeutic enzyme for treatment of hyperuricemia and gout. Now let's have a look at the aims and objectives of the study. First, I will be speaking about the process I followed to isolate uricase-producing strains of clinical Pseudomonas and environmental Bacillus. Then I will be focusing on the identification of strains with the highest uricolytic activity. Next, I will be talking about the enzyme production and uricase assay. After that, I will be explaining how I optimize the enzyme production considering parameters such as uric acid concentration, temperature, pH, carbon source, and incubation period. Finally, I will conclude by enzyme purification. For the current study, I gathered 20 strains of Pseudomonas aeruginosa from a hospital and cultured them on citramide agar, which is used to identify or differentiate Pseudomonas aeruginosa from other microorganisms. After 24 hours of incubation at 37 degrees, the characteristic features of the colonies on the agar medium were checked, and by gram staining, the morphology of the bacteria was studied. Pseudomonas aeruginosa is gram-negative and rod-shaped. It is motile because of its polar flagella and produces water-soluble pigments which diffuse through the medium. The best known are pyocyanin, which is blue or green, pyoverdin, which is yellow or green and fluorescent, and pyorubin, which is red or brown and produced by a small proportion of strains. After making sure I had isolated pure colonies of Pseudomonas aeruginosa, I cultured all the 20 strains on nutrient agar supplemented with uric acid, and after 48 hours of incubation at 37 degrees, the uricase-producing strains were isolated based on uricolytic activity on agar plate, which was identified as zone of clearance around the colonies, as can be seen in this slide. Out of these 20 strains, only 4 were able to oxidize uric acid, and the one with the highest uricolytic activity was chosen for the rest of the study. Here you can see that the selected strain was confirmed by standard biochemical tests as Pseudomonas aeruginosa. These tests include oxidase, TSI, Siemens citrate, SIM, MR, VP, OF, urease, and catalase. Pseudomonas aeruginosa has the distinctive smell described as grape-like. It is not an active fermenter of carbohydrates and produces acid but no gas in glucose. It is lactose negative but the oxidase and catalase tests for Pseudomonas aeruginosa are positive. Also, I isolated uricase-producing Bacillus strains from active sludge by using pour plate technique and basal salt medium supplemented with uric acid, which were identified as Bacillus cereus based on standard biochemical tests. As for the pour plate technique, the active sludge was serially diluted with sterile distilled water in test tubes, from which 1 cc was poured into a plate. Then I added basal salt medium supplemented with uric acid to each plate and swirled them quickly to properly mix the diluted active sludge with the medium. The mixed medium was allowed to solidify and then incubated for 48 hours at 32 degrees. Since the grown bacteria were able to produce uricase enzyme and use uric acid as the only nitrogen source to isolate uricase producing bacillus, the individual colonies were cultured on nutrient agar repeatedly, and I finally had pure bacillus colonies. After studying the characteristic features of the colonies and the microscopic morphology of the bacteria, the standard biochemical tests identified the strain as Bacillus cereus. On nutrient agar, Bacillus cereus forms large, gray-white, granular colonies with a less wavy edge and less membranous consistency. It is gram-positive and rod-shaped with square ends. They are single rod-shaped or appear in short chains, and clear-cut junctions between the members of chains are easily visible. Endospores formation are seen within the center of the cell. Spores are oval and not swelling of the mother cell. It is motile with peritraceous flagella. As you can see on this slide, the biochemical tests to identify Bacillus cereus included starch hydrolysis tests on starch agar, lecithinase on egg yolk agar, casein hydrolysis in skimmed milk agar, catalase, nitrate reduction, gelatinase, VP, mannitol fermentation, and SIM. Then I inoculated both strains in 50 milliliters of enzyme production medium containing potassium dihydrogen phosphate, magnesium chloride, glucose, and uric acid. The medium was kept in a shaker incubator for 48 hours, and then to analyze the bacterial growth and uricase production, the absorbance was calculated by a spectrophotometer at 600 nanometers for different incubation time, including 2, 4, 6, 8, 10, 24, 28, 30, 32, 34, and 48 hours, as can be seen on the current slide. The comparison of bacterial growth curves and enzyme production for both strains of Pseudomonas aeruginosa and Bacillus cereus revealed that the uricase production started with the beginning of the log phase and reached its highest level at the start of the stationary phase. The enzymatic assay for uricase was carried out by the method described by Mahler. 75 microliters of uric acid and boric acid buffer was added to 3 milliliters of boric acid buffer of pH 9, and then 20 microliters of supernatant containing uricase, which was obtained by centrifuge, was added. For the blank, instead of enzyme solution, 20 microliters of buffer was used. The blank and the test solutions were incubated at 25 degrees for 10 minutes, after which the decrease in the uric acid concentration was measured with the aid of a spectrophotometer at 293 nanometers. The difference in absorbance of the test and blank is equivalent to the decrease in uric acid concentration during the enzyme reaction. 1 unit of enzyme activity was defined as the amount of uricase required to convert 1 micromole of uric acid into a lantoin per minute at 25 degrees and at pH 9. Then I optimized the uricase production for parameters such as uric acid concentration, temperature, pH, carbon source, and incubation period for maximum uricase production. And as can be seen in the graphs, the highest level of uricase was produced with 0.3% of uric acid for both strains. The optimum incubation time for both strains was 24 hours, and the production of uricase decreased when the incubation time was longer. Uricase production for both strains increased in line with the rise in temperature, and the highest level of production was revealed to be at 35 degrees. After that, the enzyme production dropped dramatically. However, the results for the highest enzyme production, considering the parameter of pH, differed. The optimum pH for Pseudomonas aeruginosa and Bacillus cereus were 9 and 8 respectively. Enzyme production was measured in production medium containing one of the carbon sources, glucose, sucrose, maltose, and starch, and as the graphs illustrate, the optimum carbon source for Pseudomonas aeruginosa was glucose, while for Bacillus cereus was sucrose. Then, both strains were inoculated in separate optimized production medium containing potassium dihydrogen phosphate, magnesium chloride, uric acid with different carbon sources, which was glucose for Pseudomonas aeruginosa and sucrose for Bacillus cereus. The pH was adjusted at 9 and 8 for Pseudomonas aeruginosa and Bacillus cereus respectively. Then the media were incubated for 24 hours at 35 degrees in a shaker incubator. After that, they were centrifuged at 8,000 rpm for 20 minutes at 4 degrees, and 50 milliliters of the cell-free supernatants were used as crude enzymes for the following stages of concentration and purification. Solid ammonium sulfate was slowly added to cell-free supernatants at 4 degrees with gentle stirring on ice bath, and after dissolving ammonium sulfate, the mixture was kept at 4 degrees overnight, and then centrifuged at 12,000 rpm for 30 minutes at 4 degrees. The pellets were dissolved in trith hydrochloride, then dialyzed against 2 liters of trith HCl overnight under cooled conditions. Protein concentrations were measured by Bradford method using a calibration curve established with bovine serum albumin as a standard. The assay is based on the observation that the maximum absorbance for an acidic solution of Comasi Brilliant Blue G250 shifts from 465 nanometers to 595 nanometers when binding to protein occurs. Both hydrophobic and ionic interactions stabilize the anionic form of the dye, causing a visible color change. For this purpose, 100 microliters of samples obtained after dialysis were mixed with Bradford reagent, and after 5 minutes their absorbance were measured at 595 nanometers against 100 microliters of double distilled water and compared with the standard curve for bovine serum albumin. The tables on this slide compare data on enzyme activity, total units, protein contents, total protein, and specific activity for the produced enzymes for both Pseudomonas aeruginosa and Bacillus cereus at different stages of purification. Here you can see the estimated weights of the produced enzymes by sodium dotasyl sulfate polyacrylamide gel electrophoresis or STS page. Electrophoresis is a method for separating a complex mixture of protein, and STS page is used to move charged molecules through a gel matrix by means of an electric current. In the presence of STS and a reducing agent that cleaves disulfide bonds critical for proper folding, proteins unfold into linear chains with negative charge proportional to the polypeptide chain length. Now I will just conclude by a number of suggestions for future research. Molecular identification of the uricase-producing bacteria and identification of the gene encoding uricase enzyme are highly recommended. Also, recombinant DNA technology and genetic engineering can be applied to clone the gene encoding uricase enzyme in non-pathogenic bacteria. I also suggest aiming for higher levels of uricase purification by chromatography techniques such as ionic exchange chromatography, hydrophobic interaction chromatography, gel filtration, and affinity chromatography. Ionic exchange chromatography separates different types of proteins based on their net charge. In other words, ionic interactions are the basis for purification of proteins by this technique. Proteins that have a low density of net positive charge will tend to emerge first, followed by those having a higher charge density. Hydrophobic interaction chromatography is based on hydrophobic interactions between side chains bound to chromatography matrix. Gel filtration is also known as size exclusion chromatography. This technique is generally used to separate larger proteins from smaller ones by using a minimum volume of eluate. It exhibits good sensitivity and does not lead to sample loss, mainly because the solutes do not interact with the stationary phase. Affinity chromatography takes advantage of the high affinity of many proteins for specific chemical groups, and it is considered to be the most selective chromatography technique. Affinity chromatography is known to give the purest results, and it is therefore used in completing the protein purification process. Finally, preparation of PEG uricase conjugate should be focused on. PEGylation is a successful technique to improve the biochemical and therapeutic properties of proteins and their physical and thermal stabilities. The conjugation of polyethylene glycol or PEG increases the size and molecular weight of conjugated molecule and improves its pharmacokinetics or the movement of drugs through the body and pharmacodynamics or the body's biological response to drugs by increasing water solubility, protecting from enzymatic degradation, reducing renal clearance, and limiting immunogenic and antigenic reactions. Thank you so much for your time and attention. Goodbye.